Great, thank you. My name is Pete O'Haron. I want to thank LSX for having us here. I'm also here with my Chief Scientific Officer, Tom Ickham, and he'll be splitting the presentation with me. I'd like to start with a quote from Dr. Howard Ahn. He's an esteemed clinical researcher and one of our clinicians who's worked on the project from animal studies all the way through human trials. The lack of genuine success in the current status quo treatment for degenerative disc disease can only be transformed by a leap forward in science discovery and relief through pain relief through regeneration. Financial markets reward companies who boldly lead their peer group with transcendent discoveries. We've looked and we cannot find any examples where a chemical compound shaped into a pill will cure a chronic disease. Cures for chronic disease can only come through cell therapy, gene therapy, and immunotherapy. This is kind of a look at our experience. We have 50+ years experience in clinical research, healthcare delivery, and management experience. This is the nuts and bolts. We're the first to receive FDA IND clearance for Fibroblasts and therapeutic program. We have the first off-the-shelf Fibroblast product. It's quite literally a product in a box with a barcode on it. There's only 2 cell sources you can use for regenerative medicine. You can use stem cells and you can use fibroblasts. We are the world leaders in fibroblasts. Probably the most important part of this is we've had no adverse events related to our fibroblasts. Our first human trials in degenerative disc disease showed significant pain reduction using VAS and ODI scales. Over 80% of our treated patients experienced significant pain improvement. We currently have 4 peer-reviewed articles, best in this class. And our technology is disruptive to all other technologies. It has the ability to be a front door technology for the spine. Finally, our intellectual property is best in class. We are literally the world leader in fibroblasts. Our technology can fundamentally change degenerative disc disease, can lead to a surgical approach in the OR suite to the radiology suite, can also move it from the OR suite to the anesthesiology suite. It's easy to administer, a simple injection. If you can do an epidural, you can do this procedure. Again, it's an off-the-shelf allogenic human dermal fibroblast called Cybercell. HDFs are easy to acquire and expand. You can use autologous or you can use allogenic, but the need to scale and maintain quality leads us to an allogenic product. The disc space is immunoprivileged, so it allows for an allogenic product which makes scaling the cell supply much easier and more consistent quality across the entire cell line. We've tested in the lab, we tested in 2 animal trials, and we tested in human trials. The study was a double-blind, placebo-controlled 18 patients, Cybercell plus PRP, and placebo. We wanted the study to evaluate the safety profile of allogenic human dermal fibroblasts and their efficacy. We had endpoints at 3, 6, and 12 months. When you develop a product, the most important element other than safety is how does it impact the individual patient. Our safety data, we saw no adverse events related to the study product. Only 2 events related to implantation procedure, and those were resolved with no further impact. No pneumotic changes of any time at any time were found in patients after 12 months. Again, the safety data was our best possible outcome. Our intellectual property is best in field. We started with 1 provisional patent. We've organically grown that portfolio to 58 U.S. and international patents issued and 122 international and U.S. patents pending. Our IP covers all aspects of fibroblasts, from methods and composition to cell sourcing, and we've patented many other disease paths as well, and Dr. Ickham will talk to you about that. Why do we love these fibroblasts so much? Well, stem cells are owned by about 30 different companies at our last count. Fibroblasts are owned by 1 company, Fibrogenesis. They appear to be more efficacious and more potent than stem cells. They're economically superior for scaling up. and they're safe, improving characteristics in animal and human trials. Tom?
Thank you, Pete. Hello. Is this on? Yeah. Okay. So, a little bit about how we collect these fibroblasts. They're collected from qualified donors in a non-invasive tummy tuck procedure. And the exciting thing about these fibroblasts is they multiply quickly. Average stem cells, like mesenchymal stem cells, double at a rate of 36 to 48 hours. Fibroblasts in our hands can double as quickly as 24 hours, which obviously impacts the ease of generating large supply and is more economical. The fibroblasts have superior immune modulation activity compared to mesenchymal stem cells. So while both mesenchymal stem cells and fibroblasts can be used in a universal donor manner because they're immune privileged, fibroblasts are more potent at inducing T regulatory cells, interleukin-10 production, suppressing interferon gamma, and so on. The other aspect of fibroblasts is that they produce more regenerative factors compared to mesenchymal stem cells, such as EGF, IGF-1, and VEGF. Additionally, the fibroblasts have been shown to produce have been shown to be grown under conditions that do not require as much fetal calf serum, so they're more economical to generate in large scale. And we and others have demonstrated and published that fibroblasts can be differentiated into different tissues at a higher rate of differentiation than mesenchymal stem cells. Tissues including pancreatic islets, neurons, endothelial cells, and various types of cardiac tissue. So with these exciting properties of the fibroblasts, one of the animal models that we used is the experimental allergic encephalitis model, EAE model of multiple sclerosis. In this model, we've shown that the fibroblasts can actually inhibit disease onset and actually reverse established disease more effectively on a cell-to-cell basis than mesenchymal cells. Additionally, we've shown that this is associated with reduction of pathological T cells, increase in T regulatory cells, re-stimulation of induction of remyelination, where we've shown actual oligodendrocyte proliferation in the brain, as well as stimulation of neural stem cell proliferation in the dentate gyrus. So basically, they seem to work like a supercharged mesenchymal stem cell. Another model that we've used is in the spinal disc model. The clinical data that my colleague Pete talked about, this is the preclinical data where we showed that injection of fibroblasts into a punctured disc can result in increased proteoglycan and collagen II production, reduction of TNF-alpha and inflammation, as well as increase in disc height, reduction in pain, and improved mobility. A very interesting animal model, the neuropathic pain model, we've shown improvement in that as well by injecting fibroblasts intravenously. And we've dosed 12 hours after administration. The pain, the antinociceptive effect was present. It was longer lasting than gabapentin, which was very surprising to us. And it was associated with suppression of inflammatory cytokines and increase of anti-inflammatory cytokines like IL-10. For cancer is another interesting program we have, which, as maybe you know, tumor cells are often in vivo associated with tumor-associated fibroblasts that act as a shield for the tumor. The tumor-associated fibroblasts start a paracrine network with the tumor, where the tumor produces factors that activate the fibroblasts. Fibroblasts in turn activate the tumor cells to multiply, help angiogenesis, and help protect the cancer against immune attack. We've generated a fibroblast vaccine, where we take fibroblasts, make them look like tumor-associated fibroblasts by growing them under tumor-like conditions, PGE2, interleukin-10, and hypoxia, and things like that. So we make fibroblasts antigenically similar to tumor cells, irradiate them, inject them into animals with an adjuvant, and then induce immunity to tumor-associated fibroblasts, where we've shown reduction in induction of tumor immunity, reduction of tumor-associated T regulatory cells, and increased sensitivity of the tumor, and no side effects. The other very interesting area that we're using fibroblasts in preclinical studies is angiogenesis. So in this case, as you know, angiogenesis is important in things such as limb ischemia and heart failure. And we've demonstrated that the fibroblasts stimulate a coordinated cytokine production. Also, the fibroblasts have been shown in our hands to be able to differentiate directly into endothelial progenitor cells. And we've shown in the animal model of limb ischemia, limb salvage. So a snapshot of our programs and of our pipeline. We have FDA clearance and have a double-blind placebo-controlled trial for disc degeneration. That's our lead product. We have multiple sclerosis, which I've talked a little bit about. In that case, we use a fibroblast for immune modulation. Cancer immunotherapy, we can use fibroblasts for manipulating T cells and dendritic cells. Vaccine, cancer vaccine I mentioned. Therapeutics of cancer include using fibroblasts to deliver, I hope I didn't do that, using fibroblasts to deliver oncolytic viruses and payloads to tumors. Fibroblasts, like mesenchymal cells, enjoy going to hypoxic areas. CT, that's chronic traumatic encephalopathy, we have some data on that. Type 1 diabetes, using fibroblasts to enhance islet cell engraftment. Pain I mentioned, angiogenesis I mentioned. Muscular dystrophy, fibroblasts can differentiate into muscle. And we haven't published, but we have manuscript in preparation. And also the anti-inflammatory effects of fibroblasts in regards to the MDX model of multiple sclerosis, of muscular dystrophy. And also tissue generation, fibroblasts may be used in conjunction with 3D printing. So we're very excited about our work, and I'd love to hear any questions you have. Thank you.